It talks about the structure of antibodies and antigens, and you learned about how the antibody is divided into two regions: the FAB region, which is responsible for recognizing antigens and antigenic epitopes, and the constant region, which is essentially involved in the biological activity of the antibody. In today's lecture, I'll talk about how the system generates such variability in terms of antibody recognition potential. And this involves you to recall what you learned in molecular medicine in terms of DNA transcription and translation. And so I'll also tell you about how this process evolves during the differentiation of the B cell, starting from the stem cell to a functional antibody producing cell. So this is the problem, that it's through studies in mice, it's been found that you can find as many as 10 million different B and T lymphocytes, each one making or expressing a unique antibody or a T cell receptor with the ability to recognize particular non-cell antigens. But theoretical biologists have come up with this figure of around 10 to the 11 different potential antigens, whether they food antigens, bacterial, viral, parasitic antigens that you might encounter in the environment. So the question is, how does the system generate enough diversity in its B and antibody production in T cells to recognize all of these different antigens? If you thought of one gene, one protein, in other words, if we had individual genes which coded for one unique B cell with one unique receptor, then it would mean that 60% of our genome would be dedicated to just making antibodies or T cell receptors, which is highly inefficient. So the way the system gets out of this is through this process called recombination, or using components to make up one large molecule. And to describe what this means, imagine you have a pack of cards where you know that you have 4 suits: hearts, diamonds, clubs, and spades. And imagine that you had to make one molecule having one member of each suit. So you have 5 of spades matched up to 3 of hearts matched up to 2 of diamonds, and so on. So in that way, you can have 13 times 13 times 13 times 13 combinations. And in the same manner, the gene or the genes that code for antibody molecules in the variable region are derived from these or segmented forms of particular coding areas. Remember from your molecular medicine that through the central dogma you have a gene which provides the information for transcription to to code for a functional messenger RNA. Now this is simple one gene, one protein hypothesis. But the thing is that you know that this gene isn't just one continuous sequence of bases, that this gene is actually composed of a number of exons, or coding reading frames, interspersed with these non-coding regions called introns. So during transcription, the polymerase can pick out one particular exon and match it with another exon, so you get different forms of messenger RNA read by these different combinations of these exons. Right? So in the same manner, the information coding for the variable region of the antibodies are composed of three segments or three regions called the variable, diversity, and joining region. So within the variable region, you have up to 50 different reading exons. And within the joining, you have a number represented going from D1 to N, and the same for the joining D1 to N. So when a B cell gets committed to start expressing an antibody, a surface antibody molecule, you have two enzymes called RAG1 and 2, which first involved with selecting one particular D exon and linking it up to a J exon. So in this example here, you have three potential, three B cells. In the one B cell, D1 has linked up to J1, the next one, D3, is linked up to J5, and this one, D2, is linked up to J2. This is the first selection and joining that happens. After that, the V exons are joined to the DJ combination. So here we've got V1 being selected, V2, and Vn meaning any V. Right, so now we've got a VDJ segment which codes for the variable region of a heavy chain, because light chains only involve V and J. There's no D in between. Also note that the constant region exons, they all use the same constant region exon. So in this manner, you can get a wide variety of potential antigen recognition structures through this combination of VDJ for heavy chain and V and J for light chain. That's all. No, we got to sort that. Okay, thanks. And remember that within the C region, the closest to the VDJ complex is IgM. So downstream of this, we have IgG, IgA, and so on. So basically what happens is during commitment of a precursor B cell, or a stem cell at this stage, which isn't expressing a surface antibody molecule, RAG1, RAG2 gets activated to start selecting out particular exons within the chromosome of that B cell to give you a rough copy of a template VDJ rearrangement with all of the C genes tagged to that. Right? that gets transcribed into a rough messenger RNA, a long copy of messenger RNA that has this VDJ with IgM, IgG, IgA, and so on. And then after that, you have what's called post-transcriptional modification, where you get another enzyme which nicks out the constant exons for IgG, IgA, to only leave IgM tagged onto the functional VDJ antigen binding protein or exons. So this way then you have a functional IgM molecule expressed on this precursor B cell. And this is why IgM is always the first antibody made. So this diversity is generated in this first instance through the action of this RAG1, RAG2, which selects out individual VDJ recombination. Right? So in this VDJ recombination, we're talking about this part for this heavy chain, and this part for the light chain, and this constant region, we're talking about this whole, most of this heavy chain here. Because remember, this constant region is like a skeleton or a scaffold which holds the antigen binding region, which contains the biggest variability. And remember from last week, I said that within this variable region, you have three areas, CDR 1, 2, and 3, which are called hypervariable regions, because that's where you see the greatest number of amino acids changes between different antibody molecules. That hypervariability is caused by another enzyme called terminal deoxynucleotidal transferase, or TDT. So that when the VDJ rearrangements have occurred, this enzyme inserts nucleotides, any randomly 1, 2, or 3 nucleotides, in between the D and the D and the J region. So what happens when you insert additional or delete additional nucleotides? What's going to happen to your code? You're going to change your codons. And in changing your codons means you change your amino acids. So this is the reason why you're getting such hypervariability. So this way you're randomly generating more variations in the structure. The other thing too, the other contribution to this variability is that RAG1 and RAG2, which are essentially nucleases which cause the DNA to loop, and so the adjacent D and J's come together and the outside loop gets nicked out. Now this nicking isn't precise, so the nicking might occur for 1 or 2 nucleotides. So you get another source of variability in terms of removal of 1 or 2 nucleotides in this event. So this way you're also generating more variability in the final product. Now all of this happens in a precursor B cell within the bone marrow during B cell development. And this is called a pre-B cell. So the B cell developmental process starts from a stem cell in the bone marrow, goes into a pre-B cell where RAG1, RAG2 is causing this gene rearrangement. And at this stage, these pre-B cells aren't expressing any surface antibody molecules. Once this rearrangement has occurred and a functional IgM heavy chain protein is made, it's then expressed on the surface of the cell and becomes, going from a becomes a pro-B cell. Pre and pro. It's very easy to mix these two up. So it's a favorite question to ask in the exams. Alright, so just to repeat, RAG1, RAG2 is one of the contributions to variability in terms of the selection of exons for B, D, and J. And then the next additional variability is through these random insertions or deletions of nucleotides through this enzyme terminal deoxynucleotidal transferase. And there are certain rules in this generation of this functional DNA template, in that firstly, each B cell can only produce one type of antibody molecule. So it cannot produce two different types of two different specificities. One B cell, one antibody. The other is that the expression of these antibody genes follows allelic exclusion. In other words, only your antibody genes from your mother or your father are expressed in that particular cell, not both. And through this recombination, we can see the number of variabilities we can get. So within the heavy gene locus of humans, you have 51 different variable exons, 27 diversity, and 6 joining. This combination gives around 8,000, just over 8,000 recombinations. A further contribution to variability is the fact that, remember, the light chain is composed of kappa or lambda. So whether the heavy chain is associated with a particular kappa or lambda is another degree of variability. And then the third variability is the isoform, so whether that antibody is IgM, IgG, or so on. So now we'll see how this recombination occurs as the B cell develops. So first of all, I think we covered this in the previous, the early lectures, but you're aware that you can divide lymphoid tissue into primary and secondary lymphoid tissue. And primary lymphoid tissue is essentially where your precursor B and T cells develop. For B cells, bone marrow is the nursery for the pre-B cells, and the thymus for T cells. Just as an aside, do you know why they're called B cells historically? Bursa of Fabricius, so not for bone marrow. So the bursa of Fabricius is it's like an appendix which was first found in chickens, and from this organ they discovered these B cells. So this is where the first B cells activity was seen. Obviously they're called T cells because they originate in the thymus. So within these primary lymphoid tissue, the stem cells undergo proliferation to seed new B and T cells. They undergo rearrangement of these antigen receptor genes to produce a functional surface antibody receptor or a T cell receptor. At this stage, I must mention that T cells, even though they don't have these antibody structures, they have a receptor which is also composed of B, D, and J, just like the B cell rearrangements. And also within these primary lymphoid organs, you have this process called selection, which is important. By selection, we're talking about the process to remove any maturing B or T cells which recognize self-antigen with too high an affinity. Because if they recognize too high, they're going to be autoreactive and cause autoimmunity. If they recognize self-antigen with too low an affinity, it means they have a non-functional receptor, so they might as well just be removed. And this selection process occurs through cell signaling, because the affinity of a receptor for self-antigen generates kinase activity, so chemical signals going into the nucleus to allow that cell to either survive or to die by apoptosis. So it's all, the selection process is all about how much affinity there is between the surface receptor and self-antigens being presented within the tissue or the nursery that these precursors are growing. So in terms of B cell development, in the early stages, just after gestation, the fetal hematopoiesis occurs not in the bone marrow but in the liver. And this then, as the fetus develops, the hematopoiesis shifts from the liver to the bone marrow. But within the liver, you still have in adulthood a group of cells, or B cells, which are quite distinct from those bone marrow derived B cells. And these are called B1 B cells. I'll talk about these more in a later lecture. The ones that develop in the bone marrow are the ones that we think of as our classical B cells which circulate in the blood. Right? And the development of these B cells originate, as I said, within the bone marrow from stem cells, where they go through a sequence of differentiation events, starting from a stem cell to what's called a pro-lymphocyte, and then a pre-lymphocyte, and then an immature lymphocyte, and then a mature lymphocyte. The stem cell and the pro-lymphocyte stages happen within the bone marrow. Now, in this transition from a stem cell to a pro-lymphocyte, what it means is that that stem cell becomes committed to develop within the B cell lineage. When it shifts to a pro-lymphocyte, it still hasn't yet expressed a surface immunoglobulin receptor.
It's like a light.
Sorry?
So it's like youth cell.
Yeah, it's like a fetal B cell, right? In that it's not expressing the receptor, but internally what's happening is that RAG1, RAG2 is being activated, so it's starting to rearrange the DNA within that pre-B cell of giving a functional BDJ combination. In addition, within these DNA activity that's happening in the cell, it's also causing the cell to express these two markers called CD19 and CD10. I'll talk about CD antigens later, but all you need to know here is that we recognize B cells based on the expression of these two surface proteins called CD19 and CD10. So when you see this on the surface of the cell, you know that it's been committed, or the differentiation process has started going from a stem cell into a B cell lineage. Because these two markers are expressed right through the lifespan of that cell, from this pre-B cell into a functional, mature B cell. So this is what's happening in this pro-B cell stage, where you're getting first a particular D exon joining up with a J exon, and then the next stage is that a V exon is linked up here, so now you've got a functional, in this example, D1, D2, and J1. This is all at the DNA level, right? And then tagged on adjacent to this are all of these randomly inserted nucleotides brought about by TDT. And then all of the constant region fragments, IgM, IgD, and so on. So this is your rough DNA template, which is then transcribed to give you this long primary RNA transcript, where you've got this rearranged VDJ variable coding sequence with all of these FC regions genes tagged on. And then this rough transcript undergoes post-transcriptional modification, where all of these downstream constant region genes are cleaved out, leaving you with a functional messenger RNA consisting of VDJ tagged onto IgM constant region. And this is your poly A tail. So this is why IgM is always your first antibody made in a primary response. So after this translation of this this mature messenger RNA, you get a functional protein which is then expressed on the surface of this B cell. But note here that at this stage, it's only the heavy chain, not the light chain. And at this stage, this is called a pre-lymphocyte. So when it's expressing this heavy chain only, then two new proteins start to appear. The one is this, what's called a surrogate light chain. Because remember, the light chain hasn't undergone rearrangement yet. So the messenger RNA for the light chain hasn't appeared yet, whether it's kappa or lambda. So these two innate type molecules, B, pre-B, and alpha phi, are just there to stabilize this heavy chain. But also this immature surface antibody receptor is associated with two important co-receptors called Ig alpha and Ig beta. And this is also the pattern for the T cell receptor. Now this is an important concept, that for both the B and the T cell receptor, these receptors are just recognition structures for seeing antigens. They have too short a cytoplasmic tail to transduce the signal, chemical signal, into that B or T cell to induce transcriptional changes. In order for chemical signals to be generated, they have to be associated with accessory or what are called co-receptors. In this case, for B cells, it's Ig alpha and Ig beta. For T cells, it's another complex called the CD3 antigen, which I'll talk about later. So essentially, when this surface receptor sees antigen, it undergoes a conformational change to draw in the co-receptors, which by virtue of their intracellular cytoplasmic regions can then activate kinases which cause signal transduction into the nucleus of that B cell to then make antibodies and secrete antibodies. So without this co-receptor, even if the IgM sees antigen, it's not going to do anything. It's just going to bind to antibody. So within the pre-B cell stage, then recombination of the light chains happen, whether they're kappa or lambda. And remember that in the light chain, unlike the heavy chain, you only have two segments, B and J. You don't have a D. And this is why in the textbooks, when they talk about this process, they'll write it as V bracket D bracket J to denote that within heavy chains it's VDJ, but in light chains only V and J. So now you have an immature B cell which is composed of a fully functional heavy chain and a light chain, which may be kappa or lambda, associated with this signaling co-receptor Ig alpha and Ig beta. And this is shown here. Now within, this is still in the bone marrow. Okay, so within the bone marrow you have this immature lymphocyte which is developed from this pre-lymphocyte where it's now expressing a fully functional IgM antibody. This is where selection happens now, because within the bone marrow you have these these nurse cells which are expressing self-antigens. And these immature B cells, through their receptors, are binding or sticking to these self-antigens. And if they see these self-antigens with too high an affinity, then that generates a strong signal into that B cell which can cause it to die by apoptosis. And you can understand why that's happening. Exactly, because if it recognizes self-antigen with too high an affinity, it can cause autoimmune reactions. So in this way, the system is preventing potentially autoreactive B antibodies from being formed. If it's not recognizing self-antigens at all and not receiving sufficient chemical signals, then those cells will also die. So you're removing B cells with non-functional antibodies. So like Goldilocks, only those antibodies, only those B cells that make antibodies which recognized self-antigen with just sufficient affinity to generate enough signals into the cell to survive will come out of this process. Now, those cells which bind self-antigen with high affinity have a second chance to survive. And that second chance is in the form of another enzyme which causes subtle changes to the VDJ combinations to alter the epitope recognition. And this is called receptor editing. So by removing or deleting nucleotides, it's changing the CDR3 specificity, to modify the degree of affinity for self-antigen. If those modifications aren't enough and they're still too high in affinity, the cells will die. So in conclusion, I've described the fact that we can generate what's called this B-cell repertoire, or the diversity in each of us, through combination of what's called this combinatorial diversity through the function of RAG1 and RAG2, of selecting particular VDJ exons. Secondly, to generate these random addition or deletion of nucleotides through the action of this enzyme TDT. Thirdly, through another form of combination in the association between the heavy chain with capital or lambda light chains. And then thirdly, through the selection of particular isotypes for IgM, IgG, and whatever. The more advanced form of diversity is generated in this process called somatic hypermutation, which I'll talk about in a later lecture. Right?